Hello and welcome to Genetics with Ms. Schimmel. This is lesson number 7, and I just want you to remember that a Punnett square is a tool that can be used to help you predict the probability, the likelihood, the chance of an offspring's genotype, the type of genes it gets, and its phenotype, its physical appearance. But this time we're going to be learning about the trait called codominance. By the end of the video segment today, you should be able to create and interpret a monohybrid cross with codominance on a Punnett square. Don't forget that a monohybrid cross is whenever we look at just one trait at a time. For example, height or color, that sort of thing. A super quick review. Remember, for classical Mendelian genetics, you always have something dominant over something else that is recessive. For example, in this case, can you roll your tongue or not? There is no in between. So you could be hybrid for the trait and your partner could be hybrid, and you would end up with a 1 to 2 to 1 ratio for homozygous dominant, heterozygous dominant, and only one would be purely recessive. This is the 1 to 2 to 1, 3 to 1 dominant to recessive ratio. Another quick review from incomplete dominance. Sometimes you end up with something that's intermediate, in between. For example, yellow and blue makes green. For example, yellow and red makes orange, or maybe red and blue makes purple. There is no in-between allele. However, there is something that is dominant and something else that is dominant, but they're not really dominant over each other. They're said to be incomplete. In this case, red and blue makes purple, RB or BR. Welcome to new content about codominance. So your new example here, if you want to say this out loud like me, co-bo-show. Codominant, both genotypes are expressed in the phenotypes. So both show. Say that, co-bo-show, co-bo-show. Codominant, both alleles show. Neither allele is dominant over the other, like you saw back with incomplete dominance. You show the trait even if you just get one allele. An example before was maybe we had red and white and it made pink for incomplete dominance, but now red and white makes something both red and white at the same time. It's not pink, that's incomplete dominance. But if you're red and white at the same time, that's codominance. Here are some pictures of various flowers. The first example over here shows you some red, purple, and white. Maybe in this case we're talking about multiple alleles. We'll get to that more in the future. But in this case, maybe red is dominant over white. So if you mix red and white, you get only red. In the cases of the snapdragons in the center, you're going to have red and white, but if you happen to notice there's some pink right here, there's an in-between, that's incomplete dominance. However, in the case of the petunias at the end, for codominance, both show. So in the cases of this first picture, if you take red and a white parent, red and white makes new codominant at the end, where you end up with both red and white at the same time. Co-bo-show. Codominance, both alleles show in the phenotype. So when we talk about codominance, co-bo-show, codominance, both alleles show. Neither one is completely dominant, and you don't end up with something in between. Our new example would be like if you had something curly and something straight. In this case, the hair would end up being both curly and straight at the same time. You would have both types of hair on your head, curly hairs and straight hairs. That would be an interesting hairstyle, but it could be inherited because of codominance. Remember, in the case of incomplete dominance, if you had curly and straight, it would make a wavy hair pattern. However, if you have curly and straight, you would end up with both curly and straight at the same time. Ready to try this out on a monohybrid cross? For codominance, cross the bull with the cow. We have a red one, or maybe some people call it roan, and white. Cross a red and white. What do you get as the offspring? What's the genotypic ratio? What's the phenotypic ratio? Remember that the genotypic ratio is going to either be RW or WR, and you get 4 out of 4. You get a 4 to 4 ratio. Basically, it's a 100% chance that all of the offspring look neither like the father nor the mother. They look like a new generation that hasn't existed before. And since both traits of the parent show, it's not incomplete dominance, it's not classical Mendelian genetics, it's codominance. In this case, what's the phenotypic ratio? 4 out of 4, 100% are going to be codominant, where they have both colors shown in the phenotype. Are you ready to cross two hybrids, two heterozygotes this time? What's the genotypic ratio? What's the phenotypic ratio? Figure it out. Anytime we start with two hybrid parents, we're going to end up with one that is purely, and two that are heterozygotes like the parents, and another one that is pure whatever. In this case, we have something new and different. This one would be like the red cow. These would be like the red and white cows that you see up above, and this one would be white. This is codominance. So your genotypic ratio, 1 to 2 to 1, matches your phenotypic ratio. One of these, two of those, and another different one. This slide shows you what happens anytime you take two hybrid parents for classical Mendelian genetics. You end up with the classical 1 to 2 to 1 genotypic ratio, but this translates to 3 dominant to every 1 recessive. What about if it's incomplete dominance? How would you write wavy by wavy? Wavy is the in-between. So what do you start out with as your parents? Fill in this Punnett square. If we're talking about curly and straight making wavy, this first child would be curly, the second child would be wavy, the third child would be wavy, and the fourth child would be straight. What about, though, if you have codominance, which is our new case for today? Do you end up with that 1 to 2 to 1 ratio like you did for incomplete dominance? Do you end up with that 1 to 2 to 1 ratio like you did for incomplete dominance for both genotype and phenotype? Do you know this new concept of codominance? Both show. In the case of codominance for chickens, if you take a black one, BB, with a white one, WW, you end up with what they call checkered chicken. That's because it's not white, it's not black, it doesn't make gray, but it has both black and white at the same time. It has both white and black at the same time. This is our codominant situation. So your turn. What do you get anytime you take two checkers? What happens if you cross two heterozygotes? What happens when you cross two hybrids? Show me your work on the Punnett square. For codominance, black and white makes checkered. What happens anytime you cross two checkered parents, two hybrids, two heterozygotes? The first offspring most likely would be black. The second one would be checkered. The next one would be checkered also. And the last one would be white. Do you notice that 1 to 2 to 1 ratio for the genotypes? What about the 1 to 2 to 1 for the phenotypes? This is the same as both codominance and incomplete dominance. It's a little bit of a break away from classical Mendelian genetics. Thanks for being here, and thanks for watching about codominance. Remember that codominance is when each allele is equally important and you don't end up with a phenotype that is intermediate or in between. It's not classical Mendelian genetics where something is dominant over something recessive. Don't forget to check out these vocab words on your Quizlet. Thanks for being here.